in my lab, we're interested in self-assembly and particularly how it's controlled in living systems and in non-equilibrium environments to be controlled to happen in the right time in the right place for proper function. And so just let me actually, sorry, move this out of the way. And actually feel free to interrupt and ask questions. This is a picture of a cell, and I want to highlight the types of a few examples. Let's see if I can get this to work. Progress. Okay, hold on. There we go. So the Philopodia are driven by self-assembly of the actin cytoskeleton into these filaments. For the cell motility, actin, again, I have two actin examples here, form this branch network that's important for the cell motion. An example of self-assembly that's essential for cell biology that is have been of long interest to my lab is clathrin-mediated endocytosis. And what I'm showing here, it's at a smaller length scale. You form these very small vesicles. It's an essential pathway to transport material into the cell. So things like iron have to be taken up through protein receptors. It's also a way that we clear receptors off of the membrane. For example, after neurotransmitters are released and the receptors have to be internalized. And so, and what I'm showing here, and can you see my cursor? This little black trimer structure is clathrin. It assembles this, actually it's a soccer ball lattice. Here's another example of self-assembly, this dynamic here. So the reshaping of the membrane is something we're interested in, is driven by self-assembly. Okay, let me get the slides. Okay. But self-assembly is actually really quite ubiquitous throughout living systems and in physics and chemistry. And there's many ways you can imagine classifying different assemblies. And so I have two, what's helpful for my talk here is to think about two different types of assembly. And one is the ribosome, and one is as one representative class, and the other is the clathrin cage, which remodels the membrane, again, for this essential receptor uptake. And so depending on what type of thing you're trying to assemble, there's different challenges. So the ribosome for me is an example of very stable complex. So the assembly itself is really like a means to an end, right? You've got to form this ribosome. We've got tens of thousands in our cells because it's a machine that has to function. So somehow you've got to take all of these subunits, which are transcribed one at a time, right, and get them together. Assembly is really a means to an end. And so if this is your assembly process, one particular challenge that I want to introduce is that to get from all of these monomers to this final structure, you have to avoid kinetic traps, which I will define, so that you can form a lot of ribosomes. You don't want to waste all of the subunits, leaving them outside of the ribosome. We want them to all end up in the right place. And so to contrast, in clathrin cage formation, for us, we think of the assembly process itself as not just a means to an end. It's actually a really essential part of the function of endocytosis. Because, for example, this is a clathrin cage that we assembled in solution. The cell never wants to assemble the clathrin cage in solution. It serves no purpose there. So the assembly actually should be tightly coupled to localization to the membrane and to sensing or capturing of the cargo. And so it's really important that the assembly actually knows when and where it's supposed to happen. So it's really part of the process, we think, rather than just, you've got to do it if you need to make a ribosome. And so this presents a different sort of challenge in sort of designing the properties of the assembly components. And to say a little bit more about endocytosis, which I'll return to in the second half of the talk, this is an image from an in vitro assembly. And you can see that there's this sort of hexagonal lattice that's formed, and then these budded vesicles on the surface of a vesicle in vitro. And so it looks actually fairly similar to the structures that you see on the plasma membrane. So this is now an electron micrograph from a cell. And you can see, again, the vesicle and the flat lattices. So they look quite similar, this reconstituted and the real system. But the actual conditions that were used to drive them are very different. And so like I mentioned, In vitro, actually, they were able to get these structures to assemble without any cargo, without any receptors, right? But the whole job is to take up the cargo. So actually getting something to assemble and even bend the membrane is actually very different than achieving control of a functional process, right? We need it to take up cargo. We need it to package molecules that are required downstream. And so how do we actually tune the assembly process itself to make sure that it's proper and not just, sort of physically possible? And just one more illustration. So this is, we did some modeling work that was published last year with a group in Australia. And what they were looking at was they were imaging a protein called dynamin that actually forms this ring at the bottom of the vesicles in order to actually cause fission. And so one thing we like to note is endocytosis is sometimes thought of as a sort of housekeeping or textbook process, but it actually has to respond very dynamically. On the left, we have the formation of these coated pits. On the right, you can see that they stimulated the cell with calcium, and there's actually a 6-fold increase in the cluster density, and it actually happens over the course of seconds and minutes. So the Clathrin system is actually capable of forming 6 times as many clusters, but it doesn't, and it responds dynamically to signals at the membrane. So it has to be a very responsive system. Okay, so that's sort of the introduction, and so for the first part, I want to emphasize this first class I mentioned, that the ribosome is sort of representative. Although we don't study the ribosome, it's actually super complicated how it assembles. So living systems are really great at getting these things to form, no matter how many subunits they have to pull together to do it. If you take all of these subunits and try to reconstitute them in a test tube, right, which is what anybody who does structural biology often tries to do, the yield is truly terrible. You get, basically no ribosomes, none of these initiation factors that are important for DNA transcription. And one of the big reasons for that is called kinetic trapping. And so what I'm illustrating here is I have all of my monomers, right? I have to get them into this 7-mer that I pictured here with these little cartoon things. But along the way, of course, you have to put everything together. And so all of these intermediates here that I've illustrated each have 6 subunits, and they're all missing 1. But what that means is none of them can interconvert with one another, so they can't combine to successfully form. And so what happens is if you run out of monomers, none of these 6-mers can complete. And so you're stuck with these whole zoo of intermediates that can no longer complete. And the only way to complete them is to wait for the monomers to fall off here, and then it can join on to the other. And that's called kinetic trapping. And I should say, in our modeling that we're doing here, we actually emphasize there's no mistakes being made. So none of the subunits are binding in the wrong place. They're all binding to their correct partners. It's just that the only reason you get trapping is because of monomer starvation. And again, this is actually very easy to do if you have lots of intermediates. Okay, so how do you avoid this? Again, cells are living systems. Self-assembly in general is non-equilibrium because you start far from equilibrium with these monomers. So you can take advantage of active processes, chaperones, enzymes, cofactors. So we studied how you could get a virus to assemble very robustly by introducing cofactors. Or you can imagine, especially if the assembly is small, like a trimer, that it's actually perfectly capable of assembling from the bulk, right? They somehow evolved to be very good at robust assembly. So what I want to tell you about from this paper we published last year is how are these protocols, how can they be designed to avoid kinetic traps and achieve very high yield? What are the constraints that really ensure that these protocols actually work, especially as you try to assemble something that's bigger and bigger, something that's approaching more like a ribosome size? And then what are the trade-offs of these different protocols? And so we published this last year with my grad student Adeep, and actually a former undergrad from my lab, Spencer, who really sort of kicked off the whole project. Okay, so again, what you guys have all been learning about in this workshop, we're interested in the dynamics. So we have to solve differential equations. So one technique is molecular dynamics, and this is used historically to study self-assembly in the chemistry and the physics community. You can also do super coarse grain molecular dynamics, but that's not what we use. Here you parameterize the interactions by some distance-dependent energy function. This is physically a more detailed, accurate representation of the way molecules interact. It's harder to connect to experiment. It's harder to expand to large length and timescales. So we parameterize our interactions via rates. And you've already heard about reaction diffusion methods, the continuum PDE methods, and we use virtual cell when we need to do PDEs. We use our NERD software when we want to do particle-based self-assembly. So if you want to actually see the structure of the components and the structure of the assemblies they form, you need to have some coarse resolution of your molecules. So that's yet another reason why you might need spatial stochastic simulations. And then if we can coarsen out the spatial part, then we again do chemical kinetics, which you're learning about in this workshop. And so for this first part, we primarily did use non-spatial simulation. So we enumerated all the possible intermediates that could be formed by these subunits and then did it non-spatially, of course, because it's much faster. And then for the second part, I'll tell you about how we are using NERDs to study the spatial aspects as well, because basically we have to have a spatial component. Okay, and so again, we're using numerical methods. Even for the trimer, if you write down these differential equations, they're coupled and they're nonlinear. So, right, you have to solve them using a computer. And so we're using these rate-based models, and we're quantifying yield and time scales. So this is just a quick example illustrating a monomer that assembles as a function of time into dimers, and then they assemble the dimers into the trimers. And so what I want to emphasize in this image here is this plateau in the yield as a function of time. And so this is a signature of kinetic trapping, because eventually this system does reach equilibrium. But what you're seeing here in this plateau region is the monomer starvation. So you formed trimers and you still have dimers, but you've run out of monomers. And so the only way to escape this trap and reach finally your equilibrium yield is for the dimer to dissociate, give you back monomers, and then you can get there. But this really delays things. And so how do we create metrics to quantify this? And then we're going to use optimization to design strategies to get around this. And so one metric that we use is quite simple. It's the time it takes to get to 95% yield. So that's this tau 95. And the other metric that we used is we call it a trapping factor. Basically, it reports on the time you entered the trap region, so it's an inflection point in this curve, and the time that you exited the trapped region or the plateau region. And so you can pick it out very easily if you take the derivative. It's just two maxima. And so the ratio of these is a very good reporter on the extent to which your system has become trapped. And you can see here what, after some optimization, we were able to take this system and eliminate the traps. So the tau 95 is much shorter and there's no more kinetic trapping. The trapping factor becomes 1 because these two timescales are equivalent. Okay, so what our starting model assumption is we're going to say we have all of these monomers initially. That's how they start. We're going to assume that all the interactions between the binding partners have the same free energy or the same KD, but that and that the binding rates are all equal. So all the on and off rates are also the same, except for the off rates can change if you have more than one bond that are breaking at a time. And so then what we're going to do is relax these assumptions and see if we can get around the problem of kinetic trapping. Oops. Okay. So I just want to briefly establish how do we get these, when is the system going to be kinetically trapped? And so this is going to happen when you start to reach high yield. So what I'm showing on the x-axis is the stability of the interactions between the subunits. And as you expect from thermodynamics, as you stabilize the interactions, the yield goes from 60% to 100%. So that's great. That's what we expect. And if you keep stabilizing it, you can't get above basically 100% yield, right? All of these subunits are in the trimer. But if you look at the time scales, as you reach right around 99% yield, the system gets faster and faster at assembling. And then what happens at time 95, this is the star here, this is the fastest possible assembly. As you overstabilize the system, your yield stops changing, but your time scales dramatically slow down. And this is kinetic trapping. So now it's going to take you very long to reach the yield because you get stuck in all these intermediates. And so the trapping factor that I defined tracks very well with this as it's a convenient metric to track with this as well. Okay, and just to illustrate for later, you can say, well, I know what you should do. You should just pick, and you can, in fact. The fastest assembly you get is if you pick this exact free energy for your interactions, and then you will have very high yield and very fast assembly. What we're going to show is this is really, that's fine, you can do that. It's not particularly robust because if you move either direction, you can slow down dramatically. What I want to emphasize is this actually requires a very close balance between assembly and disassembly. So you start to assemble, and as you form dimers, they can fall back apart. So it requires a balance between assembly and disassembly so that you can avoid getting stuck in the traps. What happens as you overstabilize is that this balance gets disrupted and now you're not disassembling fast enough. And so what we're going to show is kinetic protocols can avoid this entirely. Okay. And the last thing I want to say about kinetic trappings, you can say, well, maybe it's not so bad. It gets worse and worse the larger the assembly. So if we have 4, 5, 6, 7 subunits, this trapping cost becomes dramatically worse as you try to stabilize the system, and also becomes more sensitive. So these windows that I'm showing here are sort of the regimes where you can have fast assembly without getting trapped. And the reason for that is quite simple. As you try to attach something to something bigger, it forms more contacts, and so the intermediates are just longer lived. It takes a longer time for them to fall apart and return the monomers to your system so you can complete. And we can actually show that this time scale has universal scaling with the free energy of the interaction and the number of subunits. Okay, and the last problem with trapping is it doesn't just slow you down. You get stuck in these trap states. The yield becomes very bad. Again, the larger the assembly that you're trying to form, this is because there are many, many ways to form an intermediate when you have 7 different subunits. Okay, so it's a problem. How do we avoid it? Our goal was to optimize these assembly pathways by modifying rate constants. So we're going to keep the structure of the assembly untouched. The concentration is not going to change, and neither is the free energy. So the equilibrium we're going to reach is identical. We're just going to try to mess around with the timescales to get there faster, much faster. Okay, and so this is an optimization problem, which means that we have to find the minimum or the maximum of a function, right? And so what we want is for our yield to be very high at a time t-sim that's short, that's fast, basically. So that's our goal, and we're going to try to find that maximum by moving around in the space of rate constants. So this is our optimization problem. How do we maximize yield at a particular time? By modifying the rates. And so that means we need a gradient. We need to compute gradients. And we potentially need lots of gradients because we're going to assume that all the subunits can be different, right? The ribosome, all the subunits are different. So you have a lot of rates that you can mess around with. And so how are we going to do this? We need gradients. We're going to use a technique that's a really neat technique. It's called automatic differentiation. And so this is it's an algorithm for computing the gradient that is commonly used in deep learning. So if you guys are familiar with neural networks, the way that they get updated is by stochastic gradient descent, and the way that the gradient is computed is using backpropagation. And automatic differentiation is actually just a generalization of backpropagation. So it turns out if you can numerically solve something and all of the steps in your algorithm are differentiable, then you can compute the gradient of any algorithm. It doesn't have to be a neural network. And so that's what we're using. We're applying autodiff to our numerical integration that we're doing of our functions. Okay, so we're going to optimize yield at a particular time using autodiff. So this, you can imagine this is the yield. It depends on all of these properties. We're going to add constraints because we can't have binding that's faster than the diffusion limit. And this is our loss function now, L. And then we need to compute the derivative of the loss function with respect to the rates and follow it to the minimum. And that's what the autodiff gives us. Okay. And so that's what we did. And we, again, motivated by what real living systems do, we chose 3 different kinetic protocols. We contrast internal design. So if you say, I want to build a trimer, can you tell me what should the rates of the binding interactions be between these different subunits, right? A plus B, C plus B, such that you have very fast assembly. And so I'm the one who's designing the rates. And then we're contrasting these with external and active protocols. And so in these cases, we imagine evolution gave you a complex, right? And then you're saying, how do I get this complex to assemble in an efficient amount of time? And so we can't touch the binding rates, but we can add in enzymes that can drive disassociation and therefore sort of recycle. And we can control the concentration and time. So we can say, well, if you titrate the monomers in slowly, you can get very good assembly. And so these are the different dimensions that we tried to optimize along. And just to remind you again that the efficiency of the assembly for these systems that we generated, it's very poor because we overstabilize the delta G. So you can see these have ludicrous timescales. This is much longer than the age of the universe, trying to get these things to reach their maximal yield. And so when we use these kinetic protocols, so we define them, and then we use autodiff to say, what are the rates? They all dramatically improve the efficiency, and they retain this very high yield. And so you can see that when you give the protein to me and say, figure out what the rates of the binding interactions should be, that's actually when you get the best speed up. So these blue ones are very fast. And in fact, they agree very well with what you get if you sort of pick the perfect free energy that balanced association and dissociation. And then these external protocols, they're not quite as good, especially for the larger systems. But they do have this benefit that any evolved system that has sort of in non-optimal rates can still be gotten to assemble very efficiently by using these, so they can operate externally on an existing system. Okay, and one cool thing is that, you know, the way that we designed this optimization, all we were selecting for was speed of assembly, and yet what our kinetic protocols actually produced was solutions that were much more robust than this sort of optimal free energy one. And so the brown curve that I'm showing here is this optimal free energy that balances association and dissociation time. So if you overstabilize the system, so if you were to mutate an interface and you made everything more stable, then this one becomes very slow because you introduce kinetic trapping. But my kinetic protocols don't get any slower. They're just as fast. And what we actually were able to show that this is because what these pathways have done is they've optimized a forward pathway from monomer to dimer to trimer to tetramer without ever having to dissociate. So they actually have become completely insensitive to dissociation by sort of trimming off all of these other pathways. And so it doesn't matter even if the binding becomes completely irreversible, you can still have very fast assembly. And for the same reason, these protocols are actually typically very robust to concentration. So if you make the system all of a sudden you add in you overexpress your protein, they're still very efficient. And it's the titration protocol does suffer a little bit, and that's because it actually wants to assemble faster. You're sort of slowing it down. Okay, and so, you know, there's many things that we interrogated about what we actually learned, but I don't, you know, want to go on and on about it. What I want to say briefly is that, for example, if we look at a self-assembly of trimers, et cetera, and we have all of the diverse subunits, how did we get this to assemble? We have to create hierarchies of timescale. So these proteins interact slowly, these proteins interact fast with each other. And if you do that, what happens is you still have blue and green subunits left over that can finally add to the dimer. And so by creating this timescale hierarchy, you can get very efficient assembly. And so the ratio of these timescales is on the order of, here it's on the order of 50. The bigger the assembly, the more you have to stratify the rates, which actually makes sense because it takes longer to finish, right? If you have to keep adding in subunits, you have to wait longer before you nucleate another dimer. But the nice thing about having diverse subunits is that there's a bigger solution space as we in sort of, again, an analogy to a neural network, if you make the neural network deeper, it has more parameters, there's more there's more solutions that you can get out of it. And this is the same that's happening with this assembly system. There's more solutions if you have diverse subunits versus if all the subunits are the same. Okay, and this is something that I already said, but this is just a graph that illustrates the reason it's robust is because it becomes completely insensitive as you overstabilize the system to dissociation times. No matter how stable you make it, it doesn't care. It never has to worry about things falling apart. Okay, and then the last thing I want to say about this this part of the talk is that if biology gives you an assembly and you want to get it to assemble efficiently, you can control the concentrations as a function of time, and that's what I'm showing here. We call it the titration protocol. If you have diverse subunits, you can use a different rate for each subunit. And then again, this can allow you to assemble the system even faster. And actually, we showed that this is effectively what's happening in virus assembly, where what we're assembling here is the HIV immature virion, which is assembled inside of infected cells. And we could show it was very difficult to avoid kinetic traps and to find the sort of perfect free energy. That is not how the virus has evolved. The subunits are actually very bad at assembling until the cofactor arrives. And so this introduces an additional time scale, again, a hierarchy, and it allows it to assemble very robustly. Okay, so to conclude, the hierarchies of these timescales can make sure that your assembly is not only efficient, but it's actually very robust to perturbations of the interaction strength or concentration. And so somewhat counterintuitively, if you want to get fast assembly, you actually have to slow down some of the binding interactions, because if they're all fast, you will get kinetically trapped. The design of these pathways can really benefit from diversity in your subunits. You don't have to assert that there's any cooperativity and things getting faster as you get bigger. And these external protocols work well, even if there's nothing optimal about the binding kinetics, which is not necessarily something that evolution selects for. And then finally, this method as we show in this paper, this same framework can be used if you have experimental data and you want to learn the kinetic parameters from the experimental data, the AutoDiff framework. Okay, and so for the last part of the talk, I want to talk more about spatial control of self-assembly and this question of how clathrin-mediated endocytosis is able to assemble in a way that is coupled to membrane localization and can be tuned such that it's only really productive when it actually captures cargo, because you don't want to assemble these structures without the presence of the receptors, even though in vitro you can show physically it's perfectly capable of doing that. Okay, so and then if I have a little bit of time at the end, I'll talk about the design of an oscillator, which has been a fun project using these same methods. So you can control the timing of self-assembly if you can control their subunits, right? If you concentrate your subunits, you push them towards the product state. That's Le Chatelier's principle. So how do you increase concentration? This sort of looks like I've used a droplet. You can use droplets, but and then of course if you can reverse this process if you dilute them again. As we think about the membrane as something that effectively concentrates your subunits, and that's via dimensional reduction. So by putting them on the membrane, you've changed their concentration. They're now in units of per area. They can potentially be closer together. It's now a two-dimensional search. And so this can actually drive self-assembly, which is something that we've looked at in several papers. And so we're interested in understanding these processes when we're going to use reaction diffusion modeling, which you guys have already heard about, sort of famously dates back to Alan Turing in the 50s. He's used it to study pattern formation. And the reason it's so helpful for us, right, is we want to reach minutes or longer, microns, and then we need to be able to simulate these active systems that can take advantage of spatial gradients and chemical reactions like phosphorylation. Okay, and so we use, again, we need to do self-assembly, so we want discrete particle-based simulations. So what I'm showing here is an algorithm we developed. It's slightly different from Smolden because it's able to recapitulate the dynamics independently of the choice of the time step. It does so by using the Green's function for pairwise interaction. So you can see that It's a random walk, and when the particles collide, they can react with a rate that's specified by the user. So of course, the user specifies who interacts with who. How do we add self-assembly? We just make these rigid bodies of particles. And so unlike in spring solid, for us, these components are rigid. There's which allows you to take large time steps. And then when the two components bind, we can impose an orientation on it. And that's how we can get it to form these lattices. Okay. And so we've sort of combined all of these algorithms for 3D, 2D, 3D to 2D. Now we have an implementation for 1D and from 3D to 1D to study assembly along DNA. And so this is our software. It's called NERDS. And it's publicly, it's all open source. And this is we've applied it now to several different systems. Here's an example of our clathrin assembly on the membrane. And so I'm happy to answer any questions if you want to try using it. How easy is it to use? We've tried to make it much easier to use by making it easier to set up the models. And so this is an example where we set up the model to do virus assembly. So these structures, if you're familiar with the protein data bank, these are experimentally determined structures from cryo-ET of the HIV lattice. So this is a monomer. The monomer assembles a hexamer, and these bind to one another to form this trihexagonal lattice. And so we can take this structure and actually design the coarse components such that we can get it to assemble with the same geometry, with the same length scales. And this software we have is called IO-NERDS, and we have it's publicly available, but we're trying to actually publish this resource this spring. It's sort of been moving in fits and spurts. And we have a website now where if you have a protein and you would like to simulate its self-assembly, you can take the protein structure from the database and put it into our code, and it will give you an executable model for NERDS to actually simulate your self-assembly. Okay, and so of course this is reaction diffusion. That there's key inputs for us. You also have to specify the structure of the components, the on and off rates for all the interactions. If they move from 3D to 2D, there's a change in the rates due to the change in the dimensionality of the search, and that has to be specified. And of course, the diffusion constants for the components. The diffusion on the membrane is at least 100 times slower than diffusion in solution. Okay, so we optimized a model of clathrin assembly that was based on kinetics that had been measured experimentally. So I'm showing you here this optimized model that agrees with experiments. The clathrin's in solution. It recruits to the membrane. Clathrin actually does not bind the membrane. It's a very interesting sort of protein. It has to be recruited there by other cytosolic proteins called adapter proteins. So they actually bind to the lipids, and then the clathrin binds to them. So there's this sort of extra step, and it turns out to be very important for cargo selection. Okay, so the experimental data is shown here in black, and our model does a very nice job of reproducing the timescales. Because we know the system geometry, we can actually convert the fluorescence that they measure from arbitrary units on the y-axis to the actual density of clathrin on the membrane. And what we learned from the model is that there's two sources of cooperativity to get what was actually a relatively slow lag followed by this rapid growth of clathrin on the membrane. And one of them has been known since the 80s, that when the clathrin binds these adapters, it actually helps the clathrin assemble. So there's a cooperative strength there, but we quantified the strength. It's around 2 kT. And the other is dimensional reduction, which I already introduced, which is that your search space changes as you move from a volume search to an area search. That has units of length that has to be compensated in some way by another length scale, which measures the relative change in the binding rate from 3D. So H is the ratio of the binding rate from 3D to 2D, which also has units of length, or the equilibrium constant. Both of them have units of correspondingly volume for 3D and area for 2D. Okay, and so just a sort of a few slides on dimensional reduction. So this is a very old idea from famously Adams and Delbruck back in the 60s. If you want to find your receptor, should you do a 3D search? Or should you adsorb to the membrane and do a 2D search? So this is sort of the targeting problem. And this is been recognized for a long time, and particularly what is faster. But more recently, it's been recognized that you can actually potentially not just be faster, but be more stable. And so we generalize this problem to think about self-assembly. So you're not necessarily trying to find a target, you're trying to find your partner. And if you and your partner can both move from 3D to 2D, you can potentially have a huge gain in the reduction of search space. So your search space changes. Also, the equilibrium constant changes. And again, this has units of length. So just to say a little bit about this, it's the same for the rank. If you remember from your thermodynamics or physical chemistry class, your equilibrium constant is proportional to the free energy, the Boltzmann weighted free energy, and the volume that you measured this pairwise binding and unbinding interaction in. And so if you think about what changes, so this is the sort of a illustration of binding. You have an unbound ensemble and you bound ensemble, and you want to know the difference in free energy. There's an enthalpic contribution, there's an entropic contribution. And so what happens to these contributions when you move these components to the 2D surface? So now you have free energy, right? And it depends on the bound and unbound state. What is often assumed is that the enthalpy has not changed. So the interface is the same, but entropically the system absolutely has to change, right? You can no longer, I had a better graphic with an airplane, right? When you're an airplane and you're in 3D, you can do barrel rolls, you can do somersaults, but if you're on the membrane, you're restricted, both in the Z dimension and in terms of these orientational degrees of freedom. And so the entropy has to change. You cannot just assert that these are the same. And of course, you have to specify whatever area you're measuring this in. And so to interrogate this a little bit more, we looked at binding between two membrane-associated proteins. We used MD simulations. So we did 3D versus 2D, and we even did a pseudo 2D. So we removed the membrane and just retained all of these restrictions to orientation in Z. And one of the things that we showed is that you could assume it's purely entropic, but we showed there can also be an enthalpic contribution. And so this is basically the free energy was much lower than if it was purely entropic. And what we were able to show is that when you're on the membrane, you actually slightly change your ensemble of configurations, not just rigid body, but literally the backbone conformations. And so that's what I'm measuring here. Both of the 2D structures were actually better at maintaining this sort of compact structure that formed a very stable dimer. Okay, and so this is a measurable quantity. We're just going to assert that we know what it is. And you can think about it as a measure of how much you fluctuate when you're on the membrane. What is the length scale that you fluctuate over? So in general, we assert it's in the nanometer range. The rate constants, again, have the same length scale, but now it can also be influenced by changes to diffusion. Basically, what we're going to say is we know this. It's on the order of nanometers. Okay, and so this dimensionality factor is really going to control how much you can possibly gain by moving your components from 3D to 2D. And it's for most cells, the volume to area is a micron. H is going to be on the order of nanometers, so you can get a gain of 10 to 100, and that you can think about this as a multiple of your equilibrium constant. So like 1,000 times stronger interaction is really going to drive binding. And it's largely a concentration effect, although of course it's not completely independent of this change in the binding affinities. Okay, so that is sort of a somewhat lengthy introduction to the fact that you can trigger assembly only at the membrane, and it should be very sensitive to these dimensionality factors, right? Because you can basically make it much more likely to bind to each other if you're in this two-dimensional environment. Okay, so what I'm showing here on the left, this is my model that I already showed you. It's optimized, and then we say this model does a good job of reproducing what experimentalists measure. And then on the right, I'm showing you if we take this same model and we put it in more in vivo-like conditions. So we change the volume to area ratio. It's actually much smaller in vitro than the experiment, sorry, in vivo than the experiment, and change the concentrations to match what they are in vivo. You don't see any nucleation. And so what's going on if you just have this single adapter protein, basically there's not enough of it to recruit clathrin to the membrane, so you can take advantage of this 2D environment. So actually, the in vitro system has way too much relative adapter. The membrane is way too sticky, and that makes it very easy to recruit clathrin to the membrane. And so here we don't see any. The only way we can finally get it to nucleate these clathrin lattices if we start giving it more of these adapter proteins. So what's interesting about these simulations is every single one of these simulations has the exact same amount of clathrin. So we're not giving it more clathrin, we're giving it more adapters that can recruit it to the membrane. And that's what it needs to actually start to trigger assembly. And the more adapters you give it, the bigger it will assemble and the faster it will assemble. And what you can see here is that it's actually quite dynamic. It assembles and then it falls back apart again, which is actually what is seen in vivo. So they see that these structures can nucleate and disassemble. And so we showed that it could actually happen spontaneously if you just don't have enough links to the membrane. Okay. And so this is something that we published a few years ago. There's some limitations of the in vitro experiment and our own model parameters. So when the adapters bound to the membrane in these experiments, they did not use lipid binding. They used what are called his tags. And so they have different affinities of the proteins for the membrane. And what we know from experiment that makes it hard to interpret the in vivo experiments is they actually, in the cell, you have 1% of your lipids are PIP2. In vitro, you have to increase it tenfold. And so that's how they get these things to nucleate. So what's actually happening in vivo if this is the only way to get it to work in vitro? And can we understand that better from our model? So how much does the actual a more realistic representation of the lipid affinity, and now can we show that in vivo it is actually sensitive to cargo binding, because in those simulations we didn't give it any cargo, we were just able to get it to nucleate lattices if it had enough adapters. And so just to spend 2 minutes on this, cargo is the idea this is a receptor, right? So I have my protein on the membrane. If it binds a lipid, its off rate is just controlled by its residence time is just controlled by this off rate. If you give it lots of cargo, though, you can imagine that its residence time is going to increase, proportional in some way to the affinity for the cargo and for the number of cargo, right? If there's 0 cargo, how could it possibly stabilize you? And so we've done some analytical theory of these models to show that there's regimes where the cargo can actually really enhance your residence time. So this is the highly sensitive. And there's regimes where it doesn't do anything. Those are regimes where typically you're already very good at binding to the membrane. The same property applies to the amount of cargo. And so finally, we've been putting this together, and we actually have Sam is writing this paper now. We're hoping to submit it in the next few weeks. We can show that there's regimes where basically clathrin will not assemble without cargo. So that's the blue and the orange curves. And that's this picture here, right? You don't see any. And so here, instead of adding adapters, we added in cargo. And then he was able to get it to nucleate. And we can show that if the membrane just isn't sticky enough, then it can become very sensitive to cargo and actually nucleate, which is what we think is happening right in the cell. It's only good at nucleating when there's cargo there. Okay. And so I just want to spend 2 more minutes with this fun project we've been working on. So a lot of the 2D localization and the enhancement that you get from 2D, it requires that you stick to the membrane, right? If you can't stick to the membrane, you can't. And so you have to have lipids that you can stick to. And there's actually several biological pathways that take advantage of turnover of these lipids. So if you can convert lipids back and forth, you can change the sort of stickiness of the membrane, which is what you're seeing here in these different examples. And so we wanted to exploit that to see how that would affect recruitment and nucleation on membranes. And the key thing is that there are these 2 lipids. PIP2 is super important in endocytosis, trafficking, signaling, and it can be actively converted back to PI4P. And this lipid you can think of as sort of inert. It's not very good at recruiting stuff to the membrane. So you can go back and forth between these lipids. And this is the only lipid that's good at recruiting stuff to the membrane. So if you deplete it, right, everything's going to fall off the membrane. And so we set up a model where we added in these enzymes. And so now the amount of lipid is actually dynamically changing. And if that's all you have is a protein that binds to one lipid and then the lipids convert back and forth by these enzymes, you reach this sort of steady state. It's very boring. And but then we again took a cue from endocytosis. It turns out that these lipid enzymes bind to the adaptor proteins, and so they can get spatially localized to the membrane. That's what I'm illustrating here. So this is the 3D enzyme catalysis, and this is a 2D enzyme catalysis. So now you've got your enzyme, and it's held right at the membrane, and so it can take advantage. You effectively activate the membrane, the enzyme by sticking it to this adapter. And we make sure that our networks always obey detailed balance, except for these catalytic reactions. And so now we can get oscillations to emerge. So we can actually create negative feedback and, in fact, positive feedback to get these proteins to come on and off the membrane dynamically. And what it shows is that you can create really instabilities in your network if you can dynamically change the composition of the membrane. Of course, it's very sensitive to this dimensionality factor, right? The bigger your dimensionality factor, the more you activate, and the easier it was for us to find oscillatory solutions. And then also the bigger the dimensionality factor, the more variety of solutions. We could have very slow, very fast periods, and very large and very small amplitudes. And we could analyze, what you actually need to get the oscillations. But what I want to show you here is we can also simulate it in NERDS. And so here I'm showing the spatial model where you can see that the orange guys go onto the membrane and then they go off the membrane as you dephosphorylate these lipids. And so we can recapitulate all of these dynamics in this spatial model. It's noisier, but it's still able to reproduce the trend. And so this is something my student Jonathan has been working on for a long time, and we're also hoping to submit this in the next few months. And you can couple it to self-assembly, and therefore actually get assembly to oscillate as a function of space and time. So to conclude the whole talk, the stoichiometry and these dimensional reduction can really control self-assembly at the membrane. If you change the geometry, right, you can dramatically change what you need to actually nucleate. We think that the biochemistry is off a bit because of the cooperativity in these networks. And finally, right, so we have this simulation software for studying self-assembly. A lot of self-assembly is hard to simulate because of the span from molecular cell timescales, but we're happy to help people figure out how to use this for their own problems. And then, of course, the people who've been doing the work, a lot of what I talked about was my students Adeep and Spencer, and then Sam and Sakao have worked a lot on the classroom model. There's a bunch of undergrads here. I don't actually have a gigantic group. These are my primary students, and thank you guys for your attention.
Thank you, Margaret. And I open the discussion. So any questions? Yeah, Pedro, Les, please go ahead if you have questions. I have a question, but so I will start just to make sure. The sites diffuse, and then once they are within reaction radius, they can bind. And when they bind, they form a rigid connection between them, right? So this rigid connection can be of any length, right? I mean, of any length up to the reaction radius.
No, it can be longer than that. We don't have our molecules actually exclude volume, so they're different than Smolden in that way. So they're particles with a radius, and then there's the sum of the radius is the collision separation, and when they can reach the collision separation, a reaction can happen. And then yes, like you said, they are placed in a rigid body. They do not. It can be larger than the well, actually, yes. So it has to be exactly at the binding radius.
So here is a question. Is it exactly the sum of two binding radius? Or is it, it may be less? Because while they're within they still can continue diffusing toward each other, right?
No, no, they can't. So we have a hard, it's a real excluded volume model. They are not allowed to interpenetrate one another. So it's different
Yeah,
than
but when they interact outside
yeah,
of
when
the,
they interact, they also they can't interpenetrate. They have to, because it's important for us, we violate detail balance if when they dissociate, they're not exactly at the contact separation where they associated. So when we put them at the separation, where the collision separation, that's where we sit them when they become a rigid complex. And when they dissociate, they sit exactly at that separation again. So they never go through each other.
It's like spring salad more than Smolden.
in spring salad, they are connected with a spring at certain distance.
Yeah,
And
I mean, we have
here they are
when
kind
you
of touching. And
well when you create a single, like if you
You also have a specific distance
yeah, but so
the
that
bond is made. It's the same thing.
yeah, it's very similar, yes, in that say it's you can I mean this Clathrin model, the for each rigid body you have control over how far apart the sites are, for example. So like when the Clathrin binds another Clathrin, they sit at that radius, but the site of one Clathrin relative to another site on that same Clathrin, you have control over that.
The
You can
difference between this and spring salad is that you predefine the the shapes and the positions of the binding sites within a monomer.
Yeah, so there's orientational control in this that's rigid. Yeah,
Yeah,
you can tell it that it can be arbitrary, and then it will form a disordered lattice.
yeah.
But and so we've done that a little bit, but I know because for like droplets, they can bind to one another with multivalent molecules, but we haven't really applied it.
In spring salad, you could create like a triangle of binding sites, and that would be rigid, and then they would be able to bind to other triangles and
yeah.
that kind of thing.
But then you wouldn't be able to control how they bound to the other triangles, and so that's what you get with nerds.
So once two molecules bind in knots, they still able to rotate against each other, right?
No, no. So
They
that's
become in so you
yeah.
have and once they bind, like are they changing? Like you kind of pull one of the molecules up or down to get to the desired
Yeah, so
end.
you when they bind, you have to orient them into the proper orientation, right? Because otherwise, if you don't control that, you would never form the types of structures that are constrained by the actual molecular geometry. So yes, they can bind at any orientation, and then once they bind, you force them into the orientation you want, and then that's fixed until they dissociate.
Okay, but can it lead to conflicts when like the third molecule approaching and it's kind of it's within reaction radius, it has a probability of binding, but then it has no space to fit into this rigid structure.
So we do have steric overlap checks. So for example, if you have a Clathrin you can see that it clearly forms hexagons, right? So if you have 5 present and you try to add 2, right, because of one of the sites is available, it recognizes the steric overlap and rejects it. So it won't allow you to violate the sort of physical demands of volume exclusion. Yeah.
Yeah. Okay, thank you. More questions, please? Yeah, I mean, the Jim is asking, but Jim, you can ask, everyone can unmute yourself.
Okay. Hi, this was a fantastic talk. I'm wondering if this dimensionality factor concept might be general enough to be helpful in virtual cell or other frameworks where you could sort of use it explicitly in kinetic laws. where, especially over the years, it's been common where
Jim,
there,
you didn't
What?
listen to my talk yesterday. That's what the whole talk was about.
no, did I? Well, I apologize, Les. Okay.
Exactly. I was going to say the same thing.
All right. So, but maybe Margaret didn't hear it.
Yeah.
But anyway, is there any thoughts of having sort of a somewhat general kinetic law description that could incorporate this? And what are your thoughts on it? And unless you could pipe in too, I guess.
Well, yeah, so let me answer first. So we have been incorporating this in a lot of non-spatial models, this dimensionality factor, and there's more than one way to do it. But yes, the easiest way to introduce it is that you have all of your species that you're tracking, and you track them in units per volume. You can choose the units, right? But you want to choose one set of units because it's non-spatial. So we typically use units per volume. And then that means that whenever your species becomes restricted to the membrane and performs another interaction with something on the membrane, you have to multiply that reaction by the dimensionality factor. And so that's where it can be most simply introduced. And so you can introduce it both for the on rate and for the off rate. In terms of, for the off rate, there can be a scaling factor if you think that unbinding has a different rate in 2D or 3D. But for simplicity, we usually assume it's just the on rate that has changed as a result of the restriction. And so all of those 2D reactions are multiplied by this dimensionality factor, V over AH, right? And it's a scalar, 10, 100. And in that way, we've been able to recapitulate the spatial effects of localization that we can explicitly do in NERDS with these non-spatial models. And if there's not a huge, if it has to, we wrote a paper on this a few years ago. If there's a very large distance you have to travel to get to the membrane, then it's not as good as an assumption. And you can try to correct for the rate by taking into account how long it takes to diffuse for the surface. But I mean, if binding to the membrane is not diffusion limited, it really works very well. And we even characterized this in a paper that Jan Moraru was on a few years ago, of how effectively a non-spatial version can account for dimensionality reduction by exactly what you asked, which is introducing this dimensionality factor.
Okay, so I have to apologize to Jan also for my ignorance.
I put in the chat a link to the bioarchive of this paper that we wrote based on calibrating bimolecular reactions on the membrane using spring salad. And it does pretty much the same thing. I can't remember now, Maggie, if we referenced your paper from a couple of years ago, where now that you mention it, I do remember you're using this dimensionality factor. So if we didn't, we'll definitely put it in before we get the final paper published.
Thank you.
Hi, Jan.
Yeah, I think that someone tried to ask a question.
Yeah, there's some sort of like highly sped up.
Yeah.
I think it's Jon. Something wrong with his
Oh yeah, that would make sense.
It sounded like Mickey Mouse.
Yeah. So do we have more questions?
I don't have a question. I just have a comment, Maggie. I mean, there was particularly on the first part, that was a really great tour de force. As someone who has modeled translation, so obviously every time there's an mRNA binding the ribosome is in two parts and they have to come together, and there's all the initiation factors, and then there's the elongation factors have to be coming in and out all the time, and all of those things, all of those kinetic traps. I mean, we had to go through calibrating our model as well, and it's very, very nice that you showed this.
Yeah.
But it's the first thing that one finds out is that the rate constants cannot be anything for these things to happen. The probability of them happening by by having random values for the rate constants it's basically nil. Because you never get a trimer even. Most of the time you just get dimers, never mind trimers. So
yeah, now that you're mentioning it, I was thinking while you talked about that, we still have a few seconds before you have to leave. If they're all heterotypic binding, in other words, there aren't any homodimers or homotrimers, or do you still have to worry about kinetic traps?
Oh yeah. Yeah, for the same, it's this monomer starvation issue can happen even when they're all the same or when they're all different. It's in some ways easier if they're all the same because you have fewer parameters to work with. But yeah, as long as you have a high yield that you can achieve, then you get pushed into this trapped regime where you form these intermediates and they just can't interconvert with one another.
But I'm just trying to intuit it. If all the subunits can only assemble in one particular way, if they're all different, they're all different molecules.
That's what we assumed. That's what we assumed in those models.
So you weren't dealing, so I don't understand how you can get monomer depletion in a dimer.
You can't get it in a dimer. So the smallest one that gets trapped, if there's no mistakes, the smallest thing that can get trapped is a trimer. Because you form dimers and trimers until you run out of monomers, and then you have to wait for the dimers to fall apart.
But how can you run out of monomers if all of the subunits are different?
Because they all bind, imagine if you have 10 of each. They all bind to each other.
But that's all you need.
Yeah, no, but they form dimers and the equilibrium should be all trimers. So the point is that you will eventually get to equilibrium. So we haven't changed the equilibrium. The problem is that you are going to take a really long time to get there if you rapidly use up all of your monomers by forming all these intermediates. And now you have to wait for them to give you back your monomers.
Yeah.
Oh. So if you can have some sort of conformational change due to binding, that would also solve it probably.
Yeah, so we have this one sort of rate growth pathway where if you can slow down dimerization, and then once you form, so then that's basically creates a nucleation barrier. So the dimers are slow to form, but once they form, they rapidly finish. Then that's one way to avoid kinetic traps. But then, yeah, it typically there has to be some built-in allostery or cooperativity or even active, like if you phosphorylate a dimer very effectively and then it completes, that's another way to do it.
Yeah. But the problem with these things sometimes is that the concentrations are very different as well. I mean, that's what happens with in translation. Some of the factors are in large excess and some of them have in very, very low amounts.
Yeah, that's another.
Once you start putting that in the picture as well, it's even worse.
Yeah.
If you run out of the ones that are very low concentration very quickly, they form dimers with whatever they bind, and then suddenly there's nothing else you can do anymore.
Yeah. Right. Good point. Good point. Yeah.
Well, thank you very much. This was really, really good. It was excellent
Great.
talk.
Yeah. Thank you guys again for having me. And if anybody has any questions about the research or about trying to use nerds, I'd be happy, just email me. I'm at Johns Hopkins, so you have to do Margaret Johnson, Johns Hopkins, because Margaret Johnson is not exactly an uncommon name.
Okay, thank you very much.
All right, thank you guys.
Yeah, bye.
Bye.
Okay.